Hello, this is Mickey Urdea once again for Alteris Associates, and welcome to our next episode of Interesting Stories in the History of Diagnostics. This is going to be our third episode on cancer diagnostics. And today I'm going to begin with the various methods, and there are a lot of them, being used for the analysis of individual molecules in a variety of different applications for oncology. Last time I talked a bit about the different types of oncology tests, everything from screening, diagnosis, prognosis, staging, therapy selection and monitoring, and recurrence. All of those areas are being investigated with these new individual molecule detection methodologies. And I'm going to touch on, I think, all of them today. So let's go to that next level. Let's talk about individual molecular types that are being used in cancer. I talked in my very first episode about the use of protein biomarkers. I talked about, for instance, the Centocor markers, the alpha-fetoprotein, CEA, and now there's a new big push using new methodologies to look at more potential proteins which would be useful in cancer. Now, it may be that many of the proteins that are going to be of particular interest that have come from particular tumor cells are going to be at very low abundance. And with that in mind, the company Alamar Biosciences is using a very interesting new methodology that they call NULISA, that is going to be looking for cancer-associated proteins in very low concentration and being able to do very large numbers of them simultaneously, even thousands of different proteins simultaneously from one particular sample. It's a fascinating technology that's going to be possible to use to look at individual samples from a large number of patients using a large number of assays simultaneously and quantitating each of the proteins present. It's really remarkable what's possible. Along those same lines, the company SomaLogic has their technology called SomaScan that is currently capable of screening 7,000 proteins simultaneously for one sample. They can also quantitate those. That is also being applied to oncology. So we will see what happens with this, but it's a search capability that just has not been possible in the past. Another approach is to actually look at and actually sequence individual proteins, millions of proteins simultaneously within one sample. An example of that is the company Quantum SI, that has been developing a technology that permits them to literally sequence millions of proteins on a single chip from one sample simultaneously. They can look at the heterogeneity across the proteins. They can look at post-translational modifications. They can look at truncations. They can look at splice variants. All of this becomes possible. And looking at the heterogeneity across the protein classes. We'll see if all this pans out, but these tools are now being used to take a look at cancer. And I look forward to hearing what happens with it. Now I'm going to switch to the biggest class of new types of technologies and new types of analyses that are being done, and that's nucleic acid analysis. It's just amazing what's going on in this field. So we all know cancer cells are typically mutated. They may have one or more single nucleotide polymorphisms. They might have insertions. They might have deletions. They might have copy number variations, or even fusions. There are lots of different things that can go on. Now, if we knew the particular mutation which has occurred, it's usually possible to set up a PCR or other amplification methodology approach to analyzing the particular sample. An area where taking a look at mutations has been particularly useful has been in the area of therapy selection. It's well known that there are particular mutations which are related to particular classes of drugs. For instance, the famous KRAS mutations, EGFR, and BRAF being particularly popular. Now, often those have been analyzed using PCR-based assays. A number of different companies doing that. In fact, many companies have kits of one kind or another for that, either for their own platforms for PCR or that can be used in other people's platforms as well. I'll come back in a little while and talk about even one example of people doing point-of-care analysis for this. But another major area that's really sprung up in the last several years is going beyond simply point mutation analysis with PCR, but using sequencing, NGS. And NGS, you're going to hear me talk about NGS a lot here now, because it's being used in so many different ways. One of the early and successful approaches to the use of NGS for therapy selection was the company Foundation Medicine, which is now owned by Roche. They had set up sequencing methodologies, and they've created a series of products based on what they refer to as their Foundation One assay group. And they have been using the Foundation 1 assay to develop a series of companion diagnostics. So this would be tests associated with specific drug types. It is an LDT, but they sought FDA approval for these particular tests. So they now have several of them for very specific targeted therapies for particular cancers based on the sequences. Now, they do look at the sort of famous KRAS and EGFRs and the BRAFs, but they also are looking at over 300 additional genes. So there's a lot of information content there. Initially, most of the companies were looking at tissue samples, for instance, FFPE or formalin-fixed paraffin-embedded samples of tissue. That's where Foundation Medicine started with their program. But what's happening now is that many are switching to cell-free DNA. Cell-free DNA being, as the name suggests, independent of cells, outside of the cells, extracellular DNA in blood that can be isolated and analyzed. Now, a portion of that cell-free DNA is actually tumor-derived, and that's referred to as circulating tumor DNA, probably from circulating tumor cells. So that has opened the door for a much simpler sample type. You don't have to go back and biopsy samples if you want to do this more than once, and in some cases people do. So that now has been a transition for Foundation Medicine. And we also have the company Guardant Health. So Guardant Health also uses an NGS platform, and they started out using the cell-free DNA, and they now have approval for their so-called Guardant 360 assay, and it's approved for all solid tumor types. That's tumor profiling, but they have also sought individual companion diagnostic approvals, and they have now, I believe, 4 for selection of colorectal cancer drugs and 1 for breast cancer drugs. I mentioned earlier that there is actually activity in point-of-care diagnostics for these same types of mutations. And the company that's most advanced in that I'm aware of is Biocartis. Biocartis has a platform that has sort of large chips that they use to do the analysis. It takes approximately 4 hours to conduct, but it's a much faster process than is possible with lab-based NGS tests. They have 2 different types of their tests. One is for FFPE samples, just like everyone else, and they also now have cell-free DNA versions of their tests. Sample prep are different for the 2, so they have 2 different formats. Beyond therapy selection with NGS, there are a number of other aspects of cancer diagnostics which are being informed by NGS. A company that's doing a lot of interesting work in this area is Natera. Natera is looking at hereditary cancers, tumor profiling, but also minimal residual disease, MRD. And I think that's a particularly interesting application of their technology, and it's a series of products that they refer to as Signatera. And Signatera colorectal cancer and other types of cancers, but let me focus a bit on the work in colorectal cancer. Natera starts by getting a sample of the actual colorectal cancer tumor itself, and they do a very broad sequencing analysis on that. They take a look at the number of mutations, and this is on an individual patient basis. So looking at individual tumors. Once they've done that, they take a close look at it. They have algorithms for determining a small subset of mutations, which they can then use, usually 15 mutations. They can use those particular mutations to develop what they call a tumor-informed bespoke assay for that particular individual that can then be used to monitor those 15 mutations quantitatively in cell-free DNA as a function of time. So that can be used for looking at the effectiveness or lack thereof of the particular drug that's being used, or it can be used for looking at the recurrence of disease if it were to occur after cessation of therapy. That's very interesting to me, and I just didn't think it was going to be possible to do this in an economically reasonable fashion, but they've done it. So this is a really great example of so-called personalized medicine as applied to diagnostics. You get your own test. So I've been talking a lot about cell-free DNA, but there's also cell-free RNA. And a good example of this is the company XI Biosciences, which is looking at a series of RNAs that are usually associated with extracellular vesicles, which I'll come back to. But these are what are referred to as orphan non-coding RNAs, or oncRNAs. What they have found is that, remarkably, these RNAs seem to be almost uniquely associated with cancer patients. A large portion of the work to date has been on breast cancer because they have access to a couple of phenomenal cohorts of samples, but they are looking at a number of other cancers as well. And I think it's also surprising, there tend to be a lot of these oncRNAs, which are specific to the cancer, not just one or two. It's amazing how many. Besides simply analyzing the native sequence of RNA or DNA associated with oncogenes and with oncology, NGS is being used to look at epigenetic changes, in particular 5-methylcytosine. Now, 5-methylcytosine does occur in normal sequences, but it tends to be altered in cancer, which changes the expression level of specific genes. Now, an example of a company that's using NGS and 5-methylcytosine analysis is GRAIL, and the GRAIL Gallery assay in particular. This is an assay that is being used now for screening for cancer. They've had recent studies published which involve 15 different types of cancers, but on their website, they indicate that they're actually looking at 50 at this point. The recent studies indicate that the sensitivity is dependent, as might be imagined, on the type of cancer and also the stage of cancer. They've been seeing a range of somewhere between 43 and 50% sensitivity, and it's as high as 80% in the case of stage 4 breast cancer. So that gives you some sense of the range. That's pretty good for these screening tests. What was the biggest surprise to me is the specificity. I figured that they were going to get somewhere in the low 90s, but in fact, they've gotten over 99% specificity. Now, it's being used in patients at increased risk of cancer that are 50 years and older, and there's going to be a significant workup of those individuals after a positive test. And there are going to be some problems with false positivity, of course, even at a 99% specificity rate, but it's still pretty darn good progress. In addition to 5-methylcytosine, there's also 5-hydroxymethylcytosine and several others that I'm not going to go through today. But 5-hydroxymethylcytosine is one that's been the focus of the company Blue Star Genomics, which has recently changed its name to ClearNote. Blue Star had done quite a bit of work in pancreatic cancer. In fact, they developed a test which was submitted to the FDA, and they've received breakthrough status for that test. The company Exact Sciences also uses methylated DNA, but in a different way. They've developed a test, Cologuard, which you may know of because it's been on TV so often. Cologuard has become a standard in screening for colorectal cancer. And in contrast to GRAIL, this is being used in people who are of average risk and just 45 years and older. Quite remarkable. They use 10 DNA biomarkers and 1 protein biomarker for the test, which requires 75 grams of feces, which goes in a box. It's a pretty good size. The 2 methylated sequences are NDRG4 and BMP3. Plus, then they look at hemoglobin as their protein biomarker. The test is PCR-based mostly with the 1 immunoassay, and it's being conducted in a very large laboratory in Madison, Wisconsin. I had an opportunity to visit there in 2019, and I couldn't believe the size of this lab. They have over 300 Hamilton robots, and the trucks line up with those big boxes of feces coming in there. It's all automated processing now. It's an amazing factory. It's amazing what they've done. I don't want to leave off the use of messenger RNA quantitation, also as another nucleic acid approach to cancer diagnostics. The best example I can think of for this one is actually Genomic Health, which is now owned by Exact Sciences, by the way. But they've been around a long time. Genomic Health was one of the early pioneers in the application of nucleic acid diagnostics in cancer. Their test is based on the use of 21 messenger RNA molecules in the analysis of breast cancer samples for prognosis. There are 16 oncology-related mRNAs and then 5 so-called housekeeping genes that they use as a control. It's also a PCR-based assay, but they had to develop their own PCR methodology internally because they needed to have very exquisite precision in their assays to tell the differences in quantity of those 21 different markers. So it was a big effort on their part. If I'm not mistaken, it's been almost $120 million to do just that. The company Veracyte's another one that uses messenger RNA quantitation in cancer assays, and they use a system that's very different from PCR. It's what it's called nCounter from the company NanoString. Lastly, I'd like to talk about the in-between category I mentioned here, in between tissue, cell, and individual molecules, and in particular, the extracellular vesicles. Now, extracellular vesicles are actually liquid-bound vesicles that are excreted into extracellular space. The content of these EVs is interesting, and it's got nucleic acids, it has proteins, it has lipids, and it has metabolites, most of the components of cells. There are many different types of extracellular vesicles, one of which is exosomes. Exosomes are enclosed by single membranes, and they're excreted by virtually all types of cells. And fortunately, they're found in many diagnostic-friendly types of samples, including plasma, urine, saliva, and even tears. There's been a lot of interest in looking at exosomes in the cancer space. One example is the company Exosome Diagnostics. Exosome Diagnostics has been working on prostate cancer assays, and they actually use exosomes isolated from blood, and they analyze them by NGS and look for a series of markers within them. They're using their test to analyze samples from men that have PSA concentrations in the so-called gray zone, between 4 and 10 nanograms per mil. This helps to resolve the likely risk of developing prostate cancer in that set of individuals. The company Biological Dynamics is also working in the exosome space for oncology. They have what they refer to as the Early Cancer Detection Program, and they've been developing a laboratory-based test to look at the risk of pancreatic cancer. Now, what's interesting about their approach is it is a multi-omics approach. That is, they're not just looking at nucleic acid, but they're also looking at protein simultaneously from the same sample. In addition to the lab-based approach, they're looking at a point-of-care device that they refer to as Verita. And Verita uses a technology they refer to as ACE, and they're using this to isolate exosomes and then the nucleic acids and proteins associated with them, and simultaneously quantitating them as part of this new testing approach. So multiomics brings us way back to proteins again. And I think it's an appropriate place to stop this survey. I hope that you found the survey interesting and informative. I just, again, am amazed at the variety of things going on. Almost every week that goes by, there's something new that is reported. I think the fact is that all of these marker types are going to be useful in one way or another in cancer. The number of types is almost certainly going to expand over time. In any event, the level of effort and focus in this area is just amazing. The types of technologies, the breadth of biomarker types is just unprecedented. And I just can't wait to see where this all goes. I'm sure we're going to see applications in a number of aspects of cancer and other disease states as well, using these same approaches that will become standard of care. I hope you all enjoyed this and got something out of it. A lot of fun for me to put it all together. I couldn't believe how long the list was when I got done. And can't wait to talk more about cancer diagnostics in future episodes. PSA is coming. So this is Mickey Urdea for Alteris Associates. Thanks again for watching, and bye.